Thanks, Abby, for introducing me. Good morning, everyone. My name is Phuc Vinh Ngo, and I'm currently doing the Bachelor of Agricultural Science, Science with honor. And today I'm very excited to present my honor project with focus on assessing sensory quality of Haskap berry genotypes. And this is very lucky today is my partner industry, Howard and Denise, also attending with us.
Thank you.
But firstly, I want to take you through to the background of the Haskap berry. So the Haskap is also known as the blue honeysuckle or the honeyberry, which is originated from the cold northern region of the Canada, the Asia, and Europe. And the Haskap is they are able to grow under minus 47 degrees and considered to be the superfood because they are rich in polyphenol, particularly the anthocyanin content make up 50 to 75%, and correlated strongly with antioxidant activity. This contributes to the huge range of the health benefits. And because of this property, there is increasingly commercial interest across the world. And especially early in Australia, Tasmania Nursery has sold the Haskap as commercial plant. And when I have some trial in the southern side nursery in Tasmania, Howard that gave me some Haskap sample to taste at the liquor and the trend of that. And that was really delicious. But I have known that the taste of the Haskap is extremely sour, so the people rarely do consume as a fresh berries. Sorry, okay. Sorry guys, technical issue. Yes. The Haskap is primarily rely on the bumblebee for cross-pollination because the bumblebee are active to foray in the colder temperature compared to the other bees. The Haskap themselves vary in size and shape with a fragile skin where the anthocyanin are distributed. The breeding program across the world focus on improving flavor, the berry size, and produce the well-known shoots as the Aurora, Boreal, and Tundra. However, we don't have many research on the microclimate in Tasmania, so we don't know how to grow it well or how to produce a good quality Haskap in Tasmania. This gap in knowledge is formed a foundation for my study. The aims of my study involve in three components. Firstly, I want to investigate how the anthocyanin level vary across the genotype and the seasons. Second, I want to evaluate the electronic tone and its ability to discriminate the taste profile across the genotypes. And finally, I want to correlate the anthocyanin level with the sensory and the physical traits. From the exam, several research questions arise. Firstly, I want to determine whether anthocyanin concentration vary across 15 genotypes and to season 2024 and 2025, whether eton can effectively differentiate the taste profile across the genotype, and whether anthocyanin level correlate with the taste attribute and the berry size. My study ran from 2025 to 2026, which the Haskap sample were harvested in December of 2025 from two Tasmanian orchards, one located in the south and one located in the northern side. And the berry were then frozen for the later assessment, such as the anthocyanin analysis and sensory taste evaluation. I went to two orchards and harvested totally 15 genotypes because there is still limited research on the in the Tasmania, so there are not many established cultivars. In my study, I only include Amazon and Amber, recognized as a cultivar undergone multiple generations of breeding. So for this reason, my study only focused primarily on the genotypes rather than cultivar. The monitoring method are involved in 3 assessments. Firstly, I did the physical assessment, should at the very side. Second, I did the physical chemical, should at the anthocyanin analysis. And finally, I did the sensory taste evaluation by using the e-tongue. For the anthocyanin analysis, I have used the well-known protocol to measure the concentration, which begin with the homogenize the frozen berry and extract the solvent into our for the anthocyanin measurement using the spectral photometer. For the sensory taste assessment, 8 taste attributes were assessed by using the electronic tongue. The 8 taste including acetate, astringency, bitterness, acetate bitterness, richness, saltiness, and sourness. Now let's move on to the key result of my study. So firstly, Haskap berries size and shape vary significantly across the genotype, with the diameter range from the 9 to 30 millimeter, while the length range from 13 to 22 millimeter. For comparison, the blueberry side would have the diameter is 19.6 millimeter, while the length is 13.3 millimeter, which is more rare than Haskap berry set. For the anthocyanin analysis, the anthocyanin concentration varies significant across the Haskap genotype, the blueberry, and the blackcurrant, which show that the amber have the highest level, whereas the unknown 25 in the blueberry was one of the lowest. Because the anthocyanin is the pigment color, so we can see these different visually by using the diluted e-tongue sample, showing the amber have a darker purple color compared to the unknown 25 in blueberry. The reason for this is likely because in the blueberry we don't see much color in the flesh. The pigment is only in the skin, whereas the Haskap, the anthocyanin contained in both flesh and the skin. The seasonal effect was also observed in the genotype growth in Tasmania, and thankfully that Hobart have started frozen berry of the season 2024, so that I am able to compare the extent between 2 seasons. Which at the figure we can see 3 out of 5 genotype, including unknown 13, unknown 3, and unknown 4, were stable. However, the unknown 14 and unknown 9 show significant difference between 2 seasons. This is likely because normally we did the harvest in the late November, but this season 2025 is so cool, so we did the harvest late, but not late enough for anthocyanin accumulated. For the e-tongue, for the sensory taste result, we found that across the 8 tastes I have assessed, only 5 tastes, including sounded astringency, acetate astringency, richness, and saltiness, vary significantly across the genotypes. For the e-tongue result, we can use the radar chart to illustrate the taste profile of the Haskap genotypes, which showing that which the human tongue can perceive the taste, which showing that the dominant taste of the Haskap is the sourness, which is with the level higher than blueberry highlighted in the yellow color, but most genotype is less sour than blackcurrant. Looking ahead, when I think that when the breeding program focus on selling Haskap as a fresh berry consumption, I think it should select a genotype with less or milder sourness. Because I don't have much time to analyze the other results, so I want to focus on dominant taste of Haskap is sourness. So the level of sourness range from -6 to 2, which the amber showing the lowest level, whereas the unknown 3 and unknown 30 showing the highest one. For the correlation between anthocyanin and the taste attribute, we can see that the anthocyanin have a negative with the sourness, but positive with the richness, and adjacency. This is likely indicate that when we aim to reach the genotype with the higher anthocyanin level, the taste will be less sour, but more adjacency and richness. For the berry size, the berry diameter and the weight were negatively correlated with the anthocyanin, which means that smaller berry size likely to have the higher anthocyanin content. In conclusion, Haskap is indeed a superfood with a high anthocyanin level, with a level higher than blueberry more than 5 times. The fruit's berry size will also vary across the genotype, and each tone can effectively discriminate the taste profile. And for the seasonal variation, anthocyanin concentration depends on the specific genotype, which is unknown 9 and unknown 14. For correlation model, it's predicted that when we increase the anthocyanin concentration, the taste will likely decrease the sourness, but will increase the adjacency and richness. And for the berry and anthocyanin correlation, we can see that a smaller berry size will likely have a higher anthocyanin level. And I am confident to say that we can grow the Haskap, good quality Haskap berry in the milder climate such as Tasmania, but not good at some country because they already have a longer breeding program, such as the optimized protocol like fertilizer. For the future research, we should focus on develop the standardized protocol by using the e-tongue to capture the complete taste, such as the bitterness, aftertaste bitterness, and the umami, and also explore the interaction of the genotype, the environment, and the management practice to explore diverse climates. Finally, I want to say thanks to Howard and Denise and Professor Roger Stanley that supplied in the Haskap sample for my study. Without your contribution, my study cannot be done. And my thanks to my supervisor, Dr. Ali Ted, and now me that guided me, helped me going through the project. Also, thank Caroline to help me to the laboratory work on staff and student in Utah, my friend and my family to support me. Thank you for listening.
Thanks, Vinh. Wonderful presentation. Does anyone have any question for Vinh?
Vinh, do you think the period at which we do the picking would make a lot of variation?
Yeah, I probably think that is the seasonal effect have influence on the genotype, especially the unknown 9 and unknown 14 is the genotype which likely remaining in the late stage compared to the other taste, and it is preferred in the warmer climate compared to the other taste.
Yeah, it's much sweeter, like when it's properly developed. It was unfortunate at the time we did this picking.
Any other question? Let's start with Shane.
Probably a little bit related to that. What do you think about doing human sensory tests? Do you think that, as well as the e-tongue test, do you think that would be helpful?
Yeah, that's very interesting questions. So I probably think that the human sensory can also perceive the taste of the Haskap genotypes, but for the broader taste of the Haskap, we are quite difficult to different taste the own taste in the same time.
Yeah.
Rather than the e-tongue. Yeah.
Yes, please.
Thanks for your talk. It was excellent. Is there a broadly accepted definition of superfood?
Good point.
Sorry, can you repeat?
Is there a broadly accepted definition of superfood?
So normally the superfood is like contribute to the huge range of health benefits. So the anthocyanin is very correlated with the antioxidant activity, and antioxidant activity contribute to a huge range of benefits. So I think that is superfood.
Yes, please.
Thanks, Vin. Great presentation. And so good to see this work being done on Haskap berries. And credit to you, Howard and Denise, for all your hard work in getting these new crops into Tassie and experimenting. I think it's really wonderful. I guess my question is around these genotypes that you're talking about. And you've identified a few different cultivars that are well known. These other genotypes that you're talking about, these are different genetic lines that you're experimenting with, or are these, what are we actually talking about here specifically?
More or less, because we could only bring them in through quarantine.
Okay.
In a seed form, and they don't grow true to type from seed. So the names that we have with them is really just our names.
Sure.
But where the origination of them was a mixed bag. We didn't have any particular seed from any particular origin.
Okay. So we're not 100% sure that they're perhaps genetically different lines, but that they could possibly be.
Could
From
well.
a-
Yes, it's open pollinated. So,
Yeah.
And the amber and ametrine have been vegetatively propagated tissue culture to
Okay.
these cultivars.
Right.
So they're true to type.
Yeah, so that's where we're all going. Anything that we evaluate at harvest time on taste and yield, and anything that's significant goes to the tissue culture laboratory.
Okay. So something like this, a study is really helpful when that
Oh, yes.
might pick out
Yeah.
the most favoured one and take that further.
So there's about there's a couple of varieties that are real standouts,
Okay.
and they'll be going to the tissue culture. So it's an ongoing process of evaluating every season.
I've got one more question, and that is the risk of pollination and being reliant on bumblebees. Is that going to be an issue for this crop?
They ordinary bees and are also
Okay.
and they're
And a lot
growing
of
quite well.
native-
They're growing quite well in on the mainland now, and we're the only part of Australia with bumblebees.
Yep.
And so we think that the ordinary native bees and the introduced honeybees will be fine.
Great.
I guess we'll have some wonderful discussion outside during the morning.
there's one more
but,
question.
let's take a few more in the
Question for Vin. It's a good tactic to get the audience to talk to themselves because it means you avoid the questions. But anyway, I'm going to direct you back at Vin. What do these things taste like? I saw you talk about the different descriptions, but overall, I put one in my mouth and
The sourness.
it's very sour.
It's sour. Yeah.
They certainly were. No, don't always. No,
Some
not
of them are a
always.
little bit sweet. Howard's very passionate. On a range of scale, they're not sweet. They've definitely got the.
Yeah.
Sour.
It's a very
It does
hard.
change.
It's very hard. to
Sour
describe.
than blueberry, but higher than blueberry, but less sour than blackcurrant.
Sure. And another question for Vin. What's the potential of these as a commercial crop for Tasmania? What's the potential of these to be a new crop for Tasmania that we could build our industry on?
So at my research site, we can grow the good quality of Haskap with the high anthocyanin level, and the taste will be less sour. And I think that the potential we can grow at the fresh berry consumption, and also processed product with the nutraceutical value.
Thank you. I'll take one more here and then one online.
So is the end goal to turn it into a powder, like a nutritional supplement rather than jam?
That is being done in Canada. Sorry.
Not sure, but potentially we can apply for developing protocol for extracting into the powder.
And is the yield per plant higher than a blueberry or
Sorry, I don't have the
what? You don't know.
So just one more question online before we get the second speaker up. How does the anthocyanin content compare to international published figures? This is from Roger Stanley.
So the Canada regions is ranked from 40 to 130 milligram per gram, but for the Tasmania range from 16 to 100 milligram per gram, because Canada already have a longer breeding program.
Good. I think that's the last question, and Let's just put this together. So our next speaker is Yaming. Yaming will be speaking on evaluating algal and microbial communities for the bioremediation of acid mine drainage at Phuc River in Tasmania. Let's welcome Yaming.
Hello, everyone. My name is Yaming Wang. My project is on evaluating algal and microbial communities for bioremediation of acid mine drainage at Phuc River, Tasmania. This project is supervised by Dr. Diane Purcell, Dr. Shen Paul, and Professor John Bowman. So mining activities often produce an acid mine drainage, which is characterized by low pH and contains elevated concentrations of metals such as iron, copper, and zinc. This occurs when sulfide minerals such as pyrite are exposed during mining and react with oxygen and water, producing sulfuric acid and releasing dissolved metals into the environment. These conditions can cause severe environmental impacts, including water pollution, toxicity to the aquatic life, and long-term ecosystem degradation. In addition, once mining operations cease, active treatment of EMD might no longer be maintained. This creating ongoing environmental challenges. Therefore, there is a need of effective and sustainable remediation strategies that require minimal and long-term management. And ideally, we want to see the system can recover naturally. So one promising approach is using the algae to remediate the water. Algae are able to tolerate very harsh environments and absorb heavy metals and increase pH, making them a low-cost and environmentally friendly way to improve the water quality in mine impacted systems. Limited work have been done using algae in remediating the mining water, and we want also to know the site-specific conditions for how this would work. And therefore, this study is focused on three main objectives. First, we want to identify the algal and microbial communities that naturally occur in this mine systems. And then we want to evaluate their bioremediation potentials by monitoring how they do to, will they make any impact on the pH changes. And then we try to see, is there an optimal condition that support their growth and activity? So this is our study site at Phuc River. On the left-hand side there, it's North Dump Drain, which is characterized by heavily contaminated metal-rich water, giving its distinct orange color. And on the right-hand side, we got old tailings dam north. And this is a more, in a term, more diluted by the rainfall. And these two sides represent contrasting environments, allowing us to compare how algae respond to these different water chemistries and also the metal concentrations in two sites. So our method is we conducted field sampling in last year in November, and we collected water and algal samples from these 2 different mine sites. And these samples were then analysed for their water chemistry and also their, including their metal concentrations and nutrient concentration through EPA Tasmania. And then in the laboratory, we performed controlled experiment using the algae to see how they respond to different pH conditions. And then we also did DNA sequencing on the water using the water samples. That's with the Flinders University. So these are the field samples we collected last year. On the, we have some greenish algae and reddish algae. And these are how they look like under the microscope. And we can see the filamentous structures and some reddish particulates that might be impacted by the metals in the water. And after the 3 months enrichment of those field samples, we only left it with this unicellular green algae, and they are the only one that survived. And this is we used for our experiment. So for the experiment, we applied a range of pH ranges treatment from approximately 3 to 7, covering the both highly acid mine conditions and neutral conditions as a control. And algae were growing in 3 different media. The BG11 media is a controlled laboratory media, and for algae growth, and then we use the actual mine water from NDD and OTDN, both sites. And the experiment lasts for 10 days. We have to monitor the how much cells have been growing each day, and then all the other conditions are kind of controlled, temperature, light and dark cycle, and the light intensity. This are how they look like after 10 days of incubation. And we can see at low pH, they kind of gradually lost their color. They become very pale, but in the higher pHs, they maintain their greenish colors and even become more vivid. That's sort of that's an indication of their biomass accumulation. They're under like a healthy growing cycle. And these are the figures that showed how algal growth changes across different pH conditions in different types of media. And we can see the growth improves as the pH improves, like in the higher pH treatments like pH 6 and pH 7, they have positive growth, and then in the lower pHs like pH 5, 3, 4, they kind of gradually dying out. But pH 7 showed although pH 7 showed slightly higher growth than pH 6, but then there's overlaps in the error bias, meaning there's not much significant difference between the two. And overall, this demonstrates how that algae growth is strongly impacted by the pH, and their optimal growth is observed under neutral conditions. And also we observed that pH changes after algal growth, and most treatments showed an increase in pH after algae being growing in these treatments. And the strongest effect we observed is in OTDN. is in the OTDN water, and so where pH increases by up to four units, and yeah, both in pH 6 and pH 7 they increased dramatically, and but in the acidic conditions like pH 3 or pH 4, there's not much change, or even there's decrease in the pH. And then so what's the difference between all those types of media that we used, or we found that the mining water actually contains more metals compared to the lab media BG11, and then the site NDD had much higher metal concentrations compared to OTDN. They have around similar levels of iron, but NDD definitely have higher metal concentrations of other types of metals. And also because BG11 is a lab controlled media, it has much higher nitrogen level, and in contrast, in the NDD and OTDN, there's limited nutrients. It's nitrogen available. So we found this algae in this mining site, but the nutrients might be limited. Their overall performance in the natural systems. So overall, algal growth is strongly pH dependent, and the optimal growth is found at pH 6 to 7, and minimal growth we observed in the acidic environment. And algae activity also increased pH, demonstrating a clear positive feedback where growth drives alkalinization. And in conclusion, we can see that algae have some adaptations to this extreme environment, and probably further research is needed to assess how efficient and scalability under real field conditions.
Nice to go. All right. Any questions for Yaming? Yes. Okay, let's start with Caroline.
Yeah, I worked in the alumina refinery for about 8 years, and dealing with waste products was a massive part of the process. Am I understanding correctly that at the moment they're doing nothing to deal with their water? This is the first research that's being done, or is there something else that they're doing?
I think they applied some artificial treatments, like using the chemicals to neutralize the water, but using the this biological product like algae is quite a novel approach, I'd say.
Why did you choose 10 days for the incubations?
It's kind of practical for our length of the project, and yeah, and we and I think it's enough for the to see for the algae to grow and observe some changes in under those conditions.
I was wondering if 10 days has any relationship to the amount of time that the water spends in the stream before it flows off into the sea, or there's nothing to do with that? No?
Not necessarily, just for how we can achieve in this project.
Are we talking about stagnant pools, or are we talking about the Savage River, which does flow?
Sorry, stagnant.
Stagnant pools, as in still water.
Yeah, I would say there's limited water flow. There are some water flows, but they mainly stagnant and there's rainfalls that periodically that changes the water chemistry.
Right.
Are you finished, Mark?
I have one more, but I'll let you go.
Downside, you want, okay. Yeah, I mean, good job with your presentation. My question is regarding the biological remediation. I feel like it could be a slow process, right?
Yeah.
And your algae survived. I mean, you said the population may have gone down a bit, but it did survive at that lower pH, which is where you're sort of targeting.
Yep.
So just basically, the fact they survived is a good thing, isn't it?
Yeah.
And then they actually may potentially, over time, persist in the environment if you had the opportunity to apply it in the environment, and then they may do something like it's not an immediate response.
Yeah, so we're quite impressed that we found this algae in the field. They're naturally occurring and surviving in this acidic water around pH 3 and 3.1 to 3.5.
Yes.
So I think they're definitely adaptations to this acidic environment, and but the more neutralized the pH would definitely stimulate their growth. But
I'm just thinking the timelines potentially is slower than your 10-day trial.
Yeah.
You know?
Yeah.
And at the risk of asking a stupid question, time of year would have an effect then on algae growth, I would assume.
Oh, definitely. Like the temperature, the light and light, the light we can receive during the day, they all contribute to.
So I guess then, if a product was being released from a mine site, it would be better to release it, if it had to be released, to come out in, say, the summer months rather than the winter months.
Yeah, like summer month I would say helping their grow because the higher temperature and colder temperature kind of would slow down their growth.
I'll come back to you, but let's take questions from online, and then I'll come back to you. So, 2 questions online, Yaming. First one is, this is from Tori Clark. She is asking, what was the starting pH of the water samples from NDD and OTDN?
Yeah, from the North Dump drain is around 3.1. That's what we used in the experiment. We didn't adjust any pH. Just used pH 3.1, and for the old heating stand north, it's around 3.5. The time when we sampled.
Okay. Second question. Which sites did the 2 algal isolates that you lab cultivated come from?
They're from those 2 sites that we saw previously from Old Tailings Dam and North Dump Drain. They're naturally existing there. We didn't buy any samples from collection. They're just field samples. That's what we felt very interesting to look at those species. We try to ID what species they are. That's one of the objectives, but then found very hard to identify them just under the microscope. Maybe I would say further research would be required. Would be ideal. We do some DNA analysis on the after we isolated the algae strains and trying to compare with other strains what exactly species they are. That would be very helpful.
I guess this is for future, now that you've learnt so much about the algae. So we know at the higher pHs they take up more of the metals that are being put into the river, but then I guess the algae would die and it would just go back to the system. So how long in the long term will this help clear the rivers of the metal?
Yeah, that's a good question. So if we input this method, we have to regularly harvesting the algae from the water, and by the way, we removing the algae, we're kind of removing the metals from the system. So I would say it have to be done regularly, like monthly something. We can't just leave the algae there forever. We have to removing them constantly.
Thank you.
One more from Navi.
I have one more question. Is the idea to add the algae to these systems, or do they exist there already and we can encourage their growth?
Yeah, they exist there already, but in a very low amount. I would say if we want to do this like really in practice, we have to like accumulate, do some enrichment treatment first of all make them accumulate their biomass, and then maybe potentially we have some artificial photoreactors and run the water through, or have an artificial pond that putting the algae and water together and then remediate it. But if it's just relying on the natural process, and maybe they have something like collaborating with the bacteria and other microbial things, but this is very slow process. To make it more practical, you have to accumulate biomass first.
There's a clarification from Tori. So she's asking that, she's saying that her question is actually about the two surviving isolates, whether if both sides had the same surviving isolates.
Yeah, I would say like, because we only have the microscopic image as an evidence, so morphologically they look quite identical. There's not much difference. And if you look around like other sites in the world that in the acid mine drainage impacted area, see there's limited species that can survive in those ecosystems. So yeah, there's not huge variety we can discover in this extreme environment, I would say.
Yeah. Naomi, do you want to ask a question?
Yeah. I have lots, but I'll just stick to one.
Yeah.
If you maybe dredge out all the algae when you think it needs replacing, do you reckon they could possibly extract those heavy metals and reuse them?
Yeah.
Might be out of your field, I don't know.
Yeah, I'll see. Because they talking. So one like ideal, like one thing we would like to achieve is we first use the algae to remediate the water, and then we harvest the algae, and then maybe like artificially remove all those metals and then you could use the algae as a protein source, maybe feed to the cattle, or like produce agar or in all sorts of industrial things. So it's kind of a cycle. You could use the product you get from treating the wastewater.
Right. I think that's all for Yaming today. Let's put our hands together for a video. Very much. So I think that concludes the seminars for today. I want to thank everyone one more time for coming. We'll be back again here in October for end of the year on our seminar. So I hope that you'll be able to join us then. Thanks.